alaikum wa rahmatullahi wa barakatuh. Today we will continue with Activity 2 of Chapter 3, the Gene, Structure and Information Unit, Part 1. You will be supplied at the end of this video with Extra Sheet 7 and Summary Sheet 7. At the end of this video, you will be able to deduce that a gene is a fragment of DNA coding for a specific protein and define both terms, transgenesis and a transgenic organism. As we know, our body is built up from proteins like hemoglobin found in red blood cells, keratin, enzymes, and many others. We know that all what we have in our body is based on the genetic program that we have and which is held by the chromosomes found in the nucleus, where chromosomes are basically made up of the nucleotides A, T, C, and G. What relationship exists between DNA and the synthesized proteins? To understand this, we are going to analyze an experiment, but first of all, we are going to define a gene. Each chromosome carries several genes, and every gene is a sequence of nucleotides. Well, here the definition of a gene is not yet complete. We'll complete it after we explain the experiment of transgenesis. In this experiment, eggs are removed from a mouse, and a human insulin gene is inserted into a male pronucleus that has been inserted into one of the eggs. Here we notice that the female and male pronuclei correspond to the female and the male gametes before fusion during fertilization. Then the obtained embryo is transferred along with other embryos into a foster mother, where the foster mother is the mother that will bring up the child instead of the natural mother. And later we observe the birth of 3 mice. After searching for human insulin in the blood of these mice, 2 mice showed negative result, where human insulin was not synthesized in them, and only 1 showed a positive result, where human insulin was synthesized in it. To explain the results of the experiment, we noticed that these 2 mice didn't produce the human insulin protein, which means that the insulin gene wasn't inserted into their genome. However, this mouse showed the synthesis of insulin protein, which means that the insulin gene was inserted into its genome. Then we can conclude that the human insulin gene was responsible for the synthesis of the human insulin protein. In conclusion, each gene is responsible for the synthesis of a specific protein. Now we can define fully a gene. A gene is a fragment of DNA that codes for a specific protein. This is the mouse that showed the human insulin hormone in its blood. Then it is the mouse that had the insulin gene inserted into its genome. This mouse is described as a transgenic mouse. What do we mean by a transgenic organism? And what is transgenesis? A transgenic organism is the organism that has the foreign DNA or genes in its genome. Transgenesis corresponds to the process of introducing a novel or foreign gene into the genome of an organism. To sum up, a gene is a fragment of DNA coding for a specific protein. Transgenesis is the process of introducing a novel or foreign gene into the genome of an organism. And a transgenic organism is the one that has the foreign gene in its genome. Thanks for listening.